Good morning and good afternoon, everyone. Today I will be presenting a rapidly evolving area in microbiology and infectious disease diagnostics using next-generation sequencing or NGS for the identification of non-tuberculosis mycobacteria, drug resistant profiling, and clinical reporting. We will look at how this technology is advancing mycobacterial diagnostic. So the learning objective of this presentation is understanding non-tuberculosis mycobacteria, clinical significance and diagnostic challenges, next-generation sequencing in microbial diagnostics, principles and applications, drug resistance profiling via NGS, mechanism and detection of resistance, and translating NGS data into clinical practice, reporting, and actionable insights. NTMs are a diverse group of mycobacterial species that can cause pulmonary and extrapulmonary infections. With over 200 recognized species, NTMs are increasingly recognized as opportunistic pathogens, especially in immunocompromised individuals. Differentiating NTMs from Mycobacterium tuberculosis complex is crucial for proper treatment. However, there are diagnostic challenges of NTMs, including phenotypic similarity to Mycobacterium tuberculosis that, for example, NTMs can mimic MTBC in clinical presentation and lab tests, leading to misdiagnostics. The other issue is related to the culture-based methods. For the slow-growing mycobacteria like Mycobacterium ulcerans that grows extremely slowly, with colonies taking 6 to 12 weeks to appear on culture. It also requires low incubation temperature, about 30 to 32 degrees, which differ from the standard mycobacterial incubation protocols. And the prevalences of the NTM infections is rising globally. This means accurate and rapid diagnostic tools for NTMs are necessary. NGS offers high-resolution identification of mycobacterial species by sequencing entire genome or specific gene regions. This allows not only a species-level identification but also detection of mixed infections and novel or rare species. It brings a level of precision that traditional methods simply can't match. So typically the workflow starts with DNA extraction from culture positive or in some cases directly from clinical specimens. This is followed by library preparation, sequencing, and bioinformatics analysis. Databases like MycoDB and tools such as Kraken, Centrifuge, or BLAST are used to identify species and potential resistance genes. So what are the advantages of the NGS in NTM identifications? As this table shows, there are significant differences between traditional diagnostic methods and those based on NGS. One of them is time to result. Traditional methods like culture can take weeks. However, NGS-based methods can dramatically reduce the turnover time. Also, the actual speed depends on the lab setup. The mixed infections. Traditional tests often miss co-infections. However, NGS, by sequencing everything in the sample, can reveal complex microbial population. Regarding the novel species, NGS can even detect a species not previously described, especially when coupled with the comprehensive databases. And for the resistant profiling, while traditional phenotyping testing is slow and sometimes ambiguous, NGS allows for rapid detection of known resistance mutations, helping guide treatment earlier. However, there are challenges in NGS-based NTM identification. First of all, the NTMs are highly diverse, over 200 species, each with different drug-resistant profile. This makes diagnosis and treatment far more complex than MTB. Second, unlike TB, we don't have a comprehensive catalog of resistant mutations for NTMs. So even if we detect mutations and we often can interpret them reliably. These gaps limit standardizations and highlights the need for advanced tools like next-generation sequencing. Moreover, there are some technical barriers as challenges in NGS-based identification of NTMs. One of them is the high GC content. The high GC content of mycobacterial genomes complicates DNA extraction, sequencing, and assembly. The genome complexity of NTMs. Non-tuberculous mycobacteria have large genomes with regions of high variability and repetitive elements, making accurate assembly challenging. The other challenges are bioinformatics and database limitations. Regarding the bioinformatics expertise, NGS analysis requires trained personnel skilled in computational biology. Many tools rely on command line interface, further limiting accessibility. Moreover, there are limited reference database. Incomplete genome database for NTMs hinder species identification, especially for novel or rare strains. And finally, resistant genes database such as ResFinder are not optimized for the NTMs, and we don't have any catalog of the resistant mutations as we have for the Mycobacterium tuberculosis. Moreover, there are challenges in data analysis. For example, we don't have any unified pipeline for NTMs, like the tools that we have available for MTBC data analysis like TB Profiler and Microbe. Pipeline fragmentations. Analysts must combine multiple tools like Kraken2 for species identification, GATK for variant calling, or IGV for visualizations. So these increase the complexity and potential errors. Moreover, there are variability in resistant predictions, differences in analysis pipelines, and knowledge base result in inconsistent resistant predictions. A limited NTM-specific database. Current resistant database lack comprehensive NTM mutation catalog. In the next slides, we will review the workflow for the NGS-based drug-resistant profiling and species identification that we have implemented at our center in San Rafael Scientific Institute. So the first step in NGS analysis is quality control of the raw sequence reads. We use tools like FastQC to assess the metrics such as pair-based sequence quality, GC content, and adapter contamination. Optimal steps include trimming low-quality bases and filtering out poor-quality reads to ensure reliable downstream analysis. As shown in the chart, we aim for most bases to have high Phred score, ideally above 30, indicating accurate base calls. After quality control, the next step is genome assembly. This helps reconstruct the genome, which is especially important for complex or fragmented sequences. We typically use spades for Illumina data and tools like Canu or Unicycler for long reads. If both reads types are available, I mean short reads and long reads, hybrid assembly using tools like Fly or Unicycler can enhance the accuracy. The output, contigs or scaffolds, is then used for a species ID and identification and downstream analysis. Once we assemble the genome, we move on to annotation. This step identifies genes, rRNAs, tRNAs, and functional elements. Tools like PROCA and RAS help automate this process for microbial genomes. The result is an annotated genome that's ready for deeper analysis like phylogenetics or resistant genes detection. We often use 16S rRNA gene for bacterial identification because it's highly conserved across species, yet contains enough variability for classification. For even finer resolution, especially within the NTMs, the ITS region can provide added phylogenetic details. We extract these sequences using tools like Barnap, then run a blast search against the curated databases like EZBiocloud or simply nucleotide blast to determine the species identity. This slide shows two examples of 16S rRNA based identification. Sample 1 matched perfectly with Mycobacterium neoaurum. As you can see, there is 100% similarity and there is no variation in 16S rRNA, so allowing confident species level identification. So the final identification is Mycobacterium neoaurum. Instead, sample 2 aligns equally with multiple subspecies within the Mycobacterium avium complex. While we can identify this sample as Mycobacterium avium complex or MAC, 16S rRNA alone can't reliably resolve the subspecies. So this highlights a limitation of 16S, great for species level identification in many cases, but not always sufficient for subspecies or closely related strains. In sample 3, the isolate shows the closest match to the provisional strain labeled as LZJCS with a 99.38% similarity and 9 nucleotide differences out of 1,442 bases. So, since the top hit doesn't correspond to a well-defined species, we classify these as Mycobacterium species. Interestingly, this LZJC includes 3 deposited isolates from Cambodia, suggesting a possible novel or under-characterized species. Again, this case highlights how 16S analysis can flag potential new or unclassified taxa. So let's move to the real world. This case highlights the diagnostic challenges of mixed NTM infections. A 65 years old COPD patient continues to decline despite targeted treatment for Mycobacterium avium complex, which was identified by culture. Clinicians suspect co-infections, but a standard 16S rRNA sequencing only picks up the dominant M. avium signal, missing any minority species. So this emphasizes the limitation of 16S rRNA and need for more sensitive approaches like metagenomic NGS to uncover the mixed infections. So the next step is performing next-generation sequencing for this patient. The workflow starts with DNA extraction from the sputum sample or from the culture if we have available. And to do the NGS-based sequencing, the short read sequencing is performed using Illumina technology. And we did the quality control. So reads are filtered and trimmed to remove low quality bases and contaminations. And to do the analysis of 16S rRNA and ITS. So the 16S rRNA sequencing identifies a high abundance of MAC. However, we have seen minor reads corresponding to another species, Mycobacterium abscessus complex, but their significance is still unclear. So the next step is the genome assembly and taxonomic classification. The reads are assembled into contings using spades for genome reconstruction, and cracking 2 taxonomic tools classify the contings, confirming M. avium complex as the dominant species and detecting significant level of Mycobacterium abscessus as secondary infection. So the conclusion is that the 16S rRNA lonely is not sufficient for the proper identification of the subspecies and also for the mixed infections. So what is the next step for precise NTM identification? Core genome multi-locus sequence typing, or CGMLST. So CGMLST expands on traditional MLST by analyzing hundreds to thousands of conserved single copy genes far beyond 5 to 10 genes used in the classic scheme. This allows much higher resolution for phylogenetic analysis, outbreak tracking, and species differentiations. It focuses strictly on genes present in all strains of a group, which makes it a powerful tool for comparing closely related organisms and resolving taxonomic boundaries. So this slide outlines the workflow for applying CGMLST to NTM identification. So we start with genome assembly using tools like spades or fly depending on the read type. Then we extract the core genome using tools like Rory or Panaru. From there we perform sequence typing by extracting single copy genes and comparing them to reference database. Next, we build phylogenetic trees using aligners like MAFT and tree building tools such as FastTree or IQ-Tree. Finally, we visualize relationships with tools like ITOL to interpret a species clustering or a strain variation. So let's come back to the sample 2, that with the 16S rRNA, our initial identification was Mycobacterium avium complex, and we couldn't detect the subspecies. So this phylogenetic tree shows the placement of the sample 2, which clusters tightly with Mycobacterium avium subspecies Hominisuis. High bootstrap values confirm the robustness of this grouping. So using the CG, MLST and tree-based analysis allowed us to resolve the isolate down to the subspecies level, something that 16S rRNA alone couldn't achieve. So this applies also to the sample 3, that using the 16S rRNA, the most similar known species was these 3 samples deposited in Cambodia. So our initial identification was Mycobacterium species, and doing the phylogenetic tree that you can see here, that it does not cluster with any reference strain from known species, as we expected regarding the analysis of the 16S, because we haven't seen any hits with 100% similarity. So this suggests it may represent a novel or under-characterized species, and we therefore report it as Mycobacterium species. So our final identification is Mycobacterium species. So let's briefly look at the antibiotic resistant associated mutations in NTMs. Antibiotic resistance in NTM is complex, as mentioned previously. It involves not just genetic mutations, but also intrinsic resistant mechanism, efflux pumps, and enzymatic degradation. Unlike MTBC, which has relatively predictable resistant patterns, NTMs show wide variability across species and some species. Several factors limit our understanding. Species diversity means resistant mechanisms are highly variable. Genomic data are still lacking for many NTM species, especially rare or emerging ones. And there's a lack of standardized studies directly linking genetic mutations to the resistant phenotypes. Also resistant in NTMs is complex. A few genetic markers are well characterized. For example, for macrolides like clarithromycin, the ERM41 gene in M. abscessus subspecies abscessus cause inducible resistance. In contrast, subspecies maciliense lacks a functional ERM41 genes and remain microlith susceptible. So you can see the differences even among a species complex. Another example is the mutation in RRL gene or 23S rRNA, especially these codons A2058G or A2059G that led to high-level resistance in MAC, Mycobacterium avium complex, and M. abscessus. And for amikacin resistance, the RRS genes or 16S rRNA mutations that in the position of 1408 is a key marker seen in both M. abscessus and MAC. So these are the markers that are critical or crucial for the predicting resistance using genotyping data. And now looking at the reporting and clinical application. So a well-structured genomic report should include 4 key elements. A species identification, so accurate species or subspecies level classification for clinical clarity, resistant profiling, a clear list of mutation with the known or suspected links to drug resistance, treatment recommendations based on the standard protocols, guidance based on the drug resistant profile to inform targeted therapy, and visualization tools like iTool or IGV helps clinicians and microbiologists to interpret data more easily and make informed decisions. So here there is an example of how a complete NGS-based mycobacterial report might look. It includes patient and test metadata for clinical traceability, a species identification including relative abundance showing mixed infection with M. abscessus and M. avium, phylogenetic placement confirming taxonomic accuracy, and therapeutic guidance with primary, alternate, and monitoring recommendations tailored to the species and resistant profile. Finally, the report is authenticated by the lab, ensuring accountability and clinical utility. So the proposed workflow summary is for the NGS-based identification and drug-resistant profiling of NTMs are the sample collections from the, for example, culture, or in some cases, the patient sample, the DNA extraction, sequencing, processing of the sequence, pre-processing of the sequence data, and the species identifications, taxonomic tools, plus 16S and ITS, and plus assembly and phylogeny for the precise, for example, subspecies identification, and drug resistant profiling, alignment, variant calling, and annotation, and interpretation based on the current data that we have available to associate the mutation with the drug resistance, and data reporting and visualization. So this combined approach ensures robust, reproducible, and accurate species identification and resistant profiling, addressing the limitation of single tool methods like Kraken. And finally, the future direction for mycobacterial sequencing. To advance NTM diagnostics, we need to focus on 4 key areas: developing NTM-specific database to improve accuracy in species identification and resistant profiling, automated pipelines that streamline genome assembly, co-genome MLST, and resistant annotation into a single workflow, enhance accessibility, especially via GUI-based tools, so labs with limited bioinformatics expertise can still benefit, and global collaboration means a standardized shared database that will be critical for tracking resistant trends and improving diagnostic worldwide. And with this slide, I would like to thank you for your attention, and in case of any question we can discuss during the QA sessions.